So now we're getting to DNA replication, and this is also about PCR and proofreading, how they are divided, etc. So we talked about in the topic 10. This is topic 11 about the S phase, so S interphase, how they duplicate into two chromosomes, two sister chromatids, I should say, and they're connected at the centromere. And there's three types of models that we talked about that they came up with in history and science: conservative, semi-conservative, and dispersive. So we came to prove that semi-conservative is the correct one, and this is by the experiment methods. So experiment methods over here, there is the N15, which is a really heavy radioactive substance that they grew DNAs in here for a long time, and after I transfer into 14 and 14 for which is it's a light substance, so I think it's a gas, I'm not sure, but they newly synthesize more DNA in the N14 because of course they grow, so they're continuously growing. They will grow in the N15, they will transfer into N14, so they have some that are N14 soaked DNAs. So there's like the light and the heavy DNA, and it is helpful because afterwards we put it into a centrifuge, and we can see that at the beginning it is all condensed at the bottom, and then after that for the first replication, we see that the DNA will become in the middle, and that proves our point that it is a semi-conservative because one of them is light and the other one is heavy. Well, then what happens is that, since they're two strands and they're stuck together, they cannot leave each other, so then they will be at a middle middle heaviness. So like they're not too heavy, they're not too light, meaning that we can prove that it's a semi-conservative, meaning one strand is a parent, the other one is a daughter. So the semi-conservative model is that one of them, I can see right here red and purple, so one of them is a parent and the other one is a new strand, that is the daughter strand that was made. I think it's daughter strand, yeah. So then after that we do another centrifugation, and we also see that at the second round it will become one little bit on the top and one at the center, and this is really important because we can tell that one of the strands is completely new, meaning only light DNAs, and there are some that are still in the middle, meaning that after this process, well, what's going to happen is that they will do the same thing again. One is going to be a parent and the other one is going to be a sister, but however this one over here is just going to be a sister, well, not sister, daughter, I don't know, a new strand. So like if there's a new strand, of course it's going to be a light one. So this experiment showed that it was definitely going to be a semi-conservative, and that's how we can explain that. The process of DNA replications. So like I said, now we know that it is a semi-conservative. There's a parent. This is a parent. These are two parent strands, and they will make a replication fork, meaning this opening here, and there's a template that is the parent strand, and there's a synthesized daughter strand. Yeah, so daughter strand was the right word. They will replicate, so then they can continue replicating afterwards. So this is the centromere that they're going to be attached in between, which is not the direct the most accurate drawing, but either way. So reading is done from 3 to 5, meaning the parent is done. The parent strand is read from 3 to 5 to synthesize in 5 to 3 order, which is important. Just remember these order: read in 3 to 5. So scanning is 3 to 5, and synthesizing is from 5 to 3. Always on the three carbon, I think it was carbon, yeah. On the 3 carbon, they will be forming new nucleotides, so making a new strand. So the formation new strands are always on the 3 prime. Yeah, so there's an issue here, which is that the eukaryotic DNAs are linear. They're stranded with 2 ends, and we'll see why that is a problem. So there are 5 steps in the eukaryotic replication. There's just 5. Okay, let's just go into the first one is about separation of the strands. So helicase, SSBP, and polymerase will be the ones that are active, that are important roles in here. So first is helicase. No, I should talk about. Okay, let's first talk about OR. OR is the origin of replication, which is the starting point of replication, of breaking H bonds at in between the DNA. So on each side they will separate from each other from the initial point, so they will start spreading that way and that way, so like they will go each ways, and these are called origin of replication, and it's about breaking H bonds. So then first of all we have a DNA helicase, which melts the H bond as well. So they melt the H bond for DNA to separate, so it's to separate the strands that they are used for. So DNA helicase, and they will replicate. bubbles that expand away from the origin. So bubble meaning the OR, so the origin of replication, where bubbles are spread away from each other, so like this, and then between the strand and opening of replication. Need some water. So SSB, so the helicase is for melting H bond strands, separating them, and SSBP, single stranded DNA binding proteins, are binding proteins that will bind to the single strand DNA for them to stop renewing, so for them to not form H bonds. So SSBP for stopping them from sticking back on again, like the two strands. Topoisomerase removes the DNA twist tension by breaking the phosphodiester bond. So topoisomerase, remember, phosphodiester bonds between the DNA strands. Well, not strands, but phosphodiester bonds between nucleotides, I think. Ask that later. So either way, they will break the they will relieve tension, breaking. Yes, they do break the phosphodiester bond, and this twist of this DNA will be relieved. So now moving on, the second one is the so we have to realize that the OR has to be multiple in dividing, so that they can actually divide because they're linear. So the chromosomes are linear, unlike in plasmids, let's say that they're circular chromosomes. So if it's linear, it has to have so many origins of replications, since the dividing will not work otherwise. It's not a circle. If it's a circle, we only need probably one. I think it was one origin of replication. So the second step is the RNA primer. So primase. It's a short one. Primase is attached to the DNA synthesis to produce a small RNA primer. So it is complementary to the DNA sequence. Very important. Complementary meaning the opposite sign. So if it's 3 to 5, what they will put on here is 5 to 3. Very, very, very important. Complementary. So that's the primase. And we can come over here to read this already, but there's two parent strands. There's an OR right here. They are spreading, so there's 3 to 5 and 5 to 3, the parent strands, and they're leading strand and lagging strand. Why? Because this is the primase. Primase is the one that attached to it. So yeah, the pink one is the primase, is like the device that attaches the RNA primer. So the RNA primer is complementary, meaning it's opposite of 3, 5, 5, 3. So if it's 3 to 5, then it's a 5 to 3 on the DNA primer, and it will always add on the 3' end. Always add on the 3' end. So remember, adding is 5 to 3. 5 to 3. So from on the 3 side, they will add. What happens on the 5 side? They cannot add. So they have to add more RNA primers. They will keep adding. And these are little fragments that are going to be made from 3 to 5, 3 to 5, 3 to 5 each time. That's why it's lagging. And this one's leading because it can just go smoothly. And these lagging little ones are called Okazaki fragments. Okazaki is literally a Japanese person's name. I looked it up. It's a woman. I thought it was a man for some reason. It's a woman. She's still alive, I think. Yeah, she's still alive. And that word is quite new. Oh yeah, this is called anti-parallel. They are not in the same direction. They're anti-parallel. Now using DNA. Yeah, so polymerase 3. Polymerase 3 is the enzyme that will synthesize new daughter strands. So DNA polymerase 3. Remember DNA polymerase 3, because there's DNA polymerase 1 and 3, but 3 is for the daughter DNA. So it's literally this process right here of making new strands is done by DNA polymerase 3, which is an enzyme that synthesizes daughter strands. So it synthesizes nucleotides from the 3' side, and it's always from 5 to 3, definitely 5 to 3. Polymerization is could be continuous or discontinuous. So continuous is the one that is leading, and discontinuous is the one that is lagging. Yeah, so if it's continuous, only one primer is needed, so it will just go straight like over here, and discontinuous, they'll need numerous. Yeah, so it's until they reach the next bubble. Continuous is until they reach the next bubble, since there are many bubbles. And primase make DNA polymerase add nucleotide to primer. That's what I said. Yeah, DNA polymerase 3 will add the nucleotides to the primer. Primase makes a DNA backfill with DNA nucleotides. Stretch. What? Organism. Also, okazaki fragments are added. Yeah. So these are short. These are around 100 to 200, 150 actually, 150 to 200 DNA nucleotides long. So those are okazaki fragments, and then they will reach the second RNA primer. And then when they reach the next primers, DNA primer, they will detach. The DNA polymerase 3 will detach and stop synthesizing new strands. So now we're at step 4, replacing RNA primers with DNA. So this is all about just replacing that little guy, the red guy that we've been seeing, that primer, from replacing it by DNA. So that is done by the polymerase 1, which will replace RNA with DNA. Nuclease is to form the new bond between the cracks. So there's a crack here, right? Because if the RNA was removed by the DNA, well, the DNA is sitting there, but it's not perfectly bonded. So they will hydrolyze, phosphodiester their bond, and catalyze, which is condensation of phosphodiester bonds. So hydrolyze is to break, phosphodiester bond in the RNA primer. So they will break the RNA primer and will connect the DNA to the other nucleotides using catalyze. So catalyze condensation reaction of phosphodiester bonds. All about phosphodiester bond. Really important. DNA polymerase 1 is all about phosphodiester bonds. Again, DNA polymerase 1 taking off RNA. Nuclease is to forming the new bonds that were cracked because the crack happened. And the 5th step over here is about DNA ligase. So DNA ligase is the linkage. Okay, I think this is about, it's not about connecting yet. Yeah, it's all about just replacing the DNA. So reforming phosphodiester bond is over here only. So I mixed up again. So DNA polymerase 1 is for breaking the primer, and nuclease is to condensation reaction to make the DNA. I think, yeah, it's to form a DNA there. Pretty much sure. Yeah, it's pretty much about forming DNA here. And then DNA ligase is to catalyze phosphodiester bonds between the broken segments. Between okazaki fragments, they are joined. So this sugar phosphate backbone, the sugar phosphate backbone is made one more time. This is created one more time, once again. So now about proofreading. Proofreading is all about errors. So they're gonna be detecting errors, and proofreading can be so they can remove a nucleotide from the gene if it was a wrong one from the growing end. So the growing end meaning like the end that they were just growing, like if they were just working on it, they can remove it using exonuclease, which is I think it's a protein, I'm not sure. Exonuclease. Yeah, exonuclease protein that will read and remove a wrong DNA if it is the case. So then there are telomeres that are basically what we were talking about when we said there are some disadvantages in the chromosome being linear. If it is linear, we'll remember how we added the primers and we add like segments from 3 to 5. We can add only on the 3 side. But what happens for the segment that can no longer add a primer? That definitely will happen. Basically what happens is that we don't get to replicate these guys. So that's a problem, because if there is essential stuff, essential coding, essential genes in here, then well, of course these are not going to be copied. So they are not going to be copied. These are end of chromosome are not going to be copies. So what's going to happen? What we do to prevent, what the body does to prevent this problem is to add telomere repeats. So humans are kind of born with very long excess chromosome kind of. So these are called telomeres. the end of chromosomes that cannot be divided. So each daughter strand is shortened by 100 nucleotides around. Yeah, so each time it divides, it shortens for 100 nucleotides. So if they kept doing division without these extra strands that were prepared, then there's going to be deformation in the human body because we're going to be lacking too much essential chromosomes. So telomerase is this guy here. Telomerase is what is able to carry RNA primer with them to allow the extension of the DNA. So these are telomerase. Telomerase are able to extend those guys. So telomeres are the one that cannot be duplicated. Telomerase is the protein that will be able to prolong, add new DNA repeats to extend the strand of DNA. So new strands can be now synthesized by DNA polymerase shortened. So shortening of telomere is seen to have established aging, but they are not a mechanism that is very well represented. Yeah, we're not entirely sure, but there is a little bit of correlation between shortening of telomeres and of aging. And cancer is when they actively express too much telomerase, I think. So too much telomerase activity will lead to indefinite division, so that's why therefore cancer can happen. So now we're at the PCR polymerase chain reaction. So polymerase chain reactions are all about amplifying, which is developing small amounts of DNA that are the specific gene sequences that we are as humans are looking for, and this is to copy the specific DNA based on binding of complementary primers. So technique is all about using primers to duplicate the desired DNA, often really specific. Allows analysis of amounts of DNA, tiny amounts of DNA, forensic analysis, genetic disposal, etc. Yeah, there's many use in this technology here. Ingredients are the template of DNA of interest primers. Like I said, very important. They're gonna add a lot of primers actually. 2 pieces of short single-strand DNA to our template. Okay, they put 2 pieces. I don't know why it's 2 pieces. The TAQ polymerase, the TAC polymerase, is an enzyme that will catalyze adding of new nucleotides to the 3 end of OH of the primer. So this TAQ polymerase is the guy that will add new nucleotides to form the new daughter strand, kind of. It's called the Thermus aqueous thermophilic bacteria. It's a bacteria that can live in very high temperature, because we need to put the DNA sequence and everything in a very high temperature so that they will break the bonds, etc., so that the 2 strand will become single-stranded. So there's the deoxynucleic triphosphate mixture that we put in. I'm not sure why we put that. The M2 is a cofactor for, so these are the cofactors for the TAQ polymerase to work, so magnesium ion, and the buffer is to maintain the pH. Thermocycler is a device I want to use to heat it up. So first step is, there's 3 key steps. So there's the denaturation, annealing, and elongation. So those are the 3 steps I want to go over. The first denaturation is all about breaking the 2 strands into 1. This is at 94 degrees Celsius. DNA will denature and it becomes ssDNA, so it's single-stranded DNA, double-stranded DNA to a single-stranded DNA. They will separate. The heat will break the bond, so the H bond. This is mimicking helicase. That's why we don't need to have it in the body to actually have these organs like helicase. Well, not organs, proteins. I think helicase is also a protein. Yes, helicase is also a protein. Basically everything a protein here at this point. So they will break bonds. Yeah, so that's the breaking bond step. So annealing is the adding of DNA primers that will bind, that will anneal. Anneal is all about binding. It could be templates binding, or primers binding, DNA or RNA primer or whatever the primers adding or binding to the template. So it could be either case. But annealing is the process of short SSDN molecules that are binded to the complementary sequence of the target DNA. Yeah, so we added polymers in the ingredients right here, so they will bind to the DNA strand of interest, DNA template of interest, and these are very specific. They are complementary. They have to be specific and complementary to the DNA of interest, and this is going to be done in 40 to 65 degrees, a bit lower in temperature. And yeah, so these are possible because primers are higher in concentration than DNA, apparently. And the process is very short-lived, so it's around 20 to 40 seconds, so it is favorable. It will happen. The binding will happen. We require 2 primers, so one primer for the forward primer and the one for the reverse primer. And forward primer is the one that's pointing from 5 to 3 on the parental strand, 5 to 3. And reverse is from 5 to 3. 5 to 3. Yeah. Forward is always 5 to 3. Reverse primer is the other one. Either way, they are both pointing from 5 to 3 actually. And what they will do is they will determine the size of the fragment that's going to be amplified. So the 2 of them will be amplified in such a way. So there are 4 base in the DNA molecule. So there's the A, C, and G and T, and each probability is around is of 1 out of 4 chances of occurring. So if we do 1 out of 4 to the n power n, meaning n is the number of strands, so the number of sequence actually. The primary sequence, if it's of A, then there's 0.25 probability of being complementary and to be fitting to the parental strand. Whereas if it gets longer and longer, it will be a very, very, very small possibility that they will actually fit. So the sequences of the primers, I think this is trying to tell us, no, the base, the base sequence should be very small. Not sure totally actually. Elongation is all about catalyzing, so they will put the primer here, and the elongation is about making the new strand, so the new daughter strand. So it is at higher temperature now again, so 72 temperature, and they will not be renewing, so the DNA strands will not stick back together, and they will be synthesized. The new nucleotides will be synthesized from the TAQ DNA polymerase. So these guys, like I said earlier, will catalyze, will form new strands starting at the primer. So extension of strands from 5 to 3 direction. What? I'm not sure why it's 5 to 3 direction. Is it not 3 to 5 direction? Yeah, so 5 to 3 because yeah, so okay. So you have to kind of remember it independently maybe. It's from 5 towards 3. Not 3 to 5 in this case. 5 towards 3 of the parental strand, I think. Cycle repeats 20 to 40 times until it will reach the desired template. So the first time it replicates too long, and the more they replicate, they will become shorter. And they will become shorter until it reaches the specific desired strand sequence. Yeah, so really the heat here, the 72-degree heat, is what stops it from getting back together the two DNA strands. So every time they separate is a double amount of template DNA, because they will divide into 2 and make more. I'm not sure about the doubling here. After 35 cycles. So after how many cycles, how many strands, DNA of strands can be amplified? Well, what we have to do is 2 to the 35, because every time we double, we can see that it's to the power of 2, the 2 to the power of the number of replications. So each time will be 4, from 4 to 8, from 8 to 16, etc. Yeah, so there could be a question about how many copies of your target sequence. How many copies of your target sequence will you have after how many cycle? 2 to the power of that time of cycles. So cycles always to the power of the 2. Yeah, this is kind of the same principle. Yeah, so like I said, first is 2, second is 4 molecule, etc. And after 25 to 30 rounds, did not write it down. After 25 to 30 rounds, around there's millions and billions of molecules, where 99.999% is the desired molecule. So after many times replication, they will become the desired molecule. I don't know why either. Okay, that's it for